Hello dear friends, welcome back again to another video from my YouTube channel. Dear friends, in this video we are going to understand about one more very important process that is happening in the sexual reproduction in flowering plants, that is megasporogenesis. Before we discuss the entire megasporogenesis process, or before we standardize the definition of megasporogenesis, let us split the words to understand the textual meaning of the word megasporogenesis, my dear friends. Mega means large, spore means reproductive cell, and genesis means the process of formation. In short, megasporogenesis means the process of formation of megaspore. So it is the process of formation of megaspore. Now the question arises, what is this megaspore means? Megaspore means the female gametophyte. Megaspore means female gametophyte. And most of the students, they got confused between gametophyte and the gamete. Gametophyte is not a gamete, my dear friends. Gametophyte means it is a structure in which the gametes are present. For example, what is the name of male gametophyte in the angiosperm? It is the pollen grains. Pollen grains are not actually the male gametes. Pollen grains are the male gametophytes in which we have the male gamete. Similarly, the female gametophyte is nothing but a structure in which the female gamete is located, in which the female gamete is present. Now, the male gametophyte's name is pollen grain. Now, what is the name of female gametophyte? It is embryo sac. Embryo sac. Embryo sac is a female gametophyte in which the female gamete is located. In this entire process, we are going to understand how this embryo sac is getting developed, how this female gametophyte is getting formed inside the ovule. To understand that, let us recall the structure that we have studied in the previous class, that is nothing but the anatropous ovule. Anatropous ovule is covered with two protective layers. These two protective layers are called as outer integuments and the inner integuments. When the ovule is young, that means still the ovule is not yet matured. When the young ovule is there, in that young ovule, there is no development of this embryo sac. Embryo sac get developed only when the ovule become mature. Unless and until there is no maturity in the ovule, there is no development of the embryo sac inside the ovule. Now, this is the ovule in young condition, at its young condition. Now, there is no embryo sac, but we have got the outer integument as well as inner integuments. We have got hilum, we have got funicle, we have got nucellus membrane. And just recall the nucellus. Nucellus is nothing but a mass of parenchymatous cells in which abundant amount of food is reserved, rich amount of nutritive material is stored. That tissue is called as nucellus. Now, in this young ovule, we don't have any embryo sac development. We'll understand how the embryo sac is getting developed inside this young ovule. Here we have got an pore kind of thing, opening kind of thing. This opening, it is called as a micropylar end. Opposite to that micropylar end, we have the chalazal end. We made it very clear in our previous video. Now, they are all the nucellar cells. They are all the nucellar cells. Nucellar cells are red. I have labeled it in the red color. I have drawn it in the red color. Nucellar cells are abundant in their nutritive material. They are rich in the food. Any one of the cell towards the micropylar end, any one of the cell towards the micropylar end get differentiated into megaspore mother cell. Say for example, this cell get differentiated itself into a megaspore mother cell and it is towards the micropylar end. Now what has happened here? As the ovule matures, as the ovule starts maturity, starts getting mature, a nuclear cell towards micropylar end differentiated into megaspore mother cell. It is MMC, megaspore mother cell. This is the first event takes place when the young ovule is getting matured. What is the first event? The nuclear cell towards the micropylar end, it get differentiated, any one of the cell, not all the cells, any one of the cell get differentiated into the MMC, megaspore mother cell. Now let us take a closer look of this area for our better understanding. If we focus this area for our better understanding, how it looks like. This is the differentiated cell. This is the differentiated cell which is diploid in its nature, and all around this cell we have the nuclear cell. Let us make it with a red color. They are all the nuclear cells. They are all the nuclear cells surrounding to this. Previously, this cell was also a nuclear cell, but as the young ovule starts maturity, as the young ovule becomes mature, the first event is any one of the cell towards the micropylar end, it get differentiated. Now this will become our MMC. Now this MMC, it divide by meiosis. It divides by meiosis. And we know meiosis is the reductional division, and meiosis always ends by forming 4 cells. Now here we have got the diploid cell. Diploid cell undergoes meiosis. That means definitely it results into the formation of 4 haploid cells, and all 4 haploid cells are linearly arranged. They are all the haploid cells. They are all haploid cells. They are all haploid cells. Why these haploid cells are formed? Due to the meiosis. Diploid megaspore mother cell undergoes meiosis and results into the formation of 4 haploid cells, and all these 4 haploid cells, they are still covered with the nuclear cells. They are covered with nuclear cells. This is the nucleus. This structure, it is called as megaspore tetrad. It is called as megaspore tetrad. Why it is called as megaspore tetrad? Because there are 4 cells which are formed, and all the cells, they are nothing but, all of them, they are MMCs. They are MMCs, megaspore mother cells. They are all megaspore mother cells because they are all the daughter cells of this megaspore mother cell. Now, what is going to happen further? We have 4 haploid cells. Out of 4 haploid cells, 3 cells they degenerate. Out of 4, 3 cells they degenerate. 3 megaspore mother cells degenerate. Now, which 3 cells get degenerate? Whether 1, 2, 3 get degenerate, or 1, 2, 3 get degenerate, or 1, 2, 3 get degenerate, which 3 cells they get degenerate? The cells which are towards the chalazal end, they get degenerate. Now, where is that chalazal end? In this diagram, this is the chalazal end, this is the micropylar end. We have taken a cell outside, that means this is the chalazal end and this is the micropylar end. Now, this is the chalazal end and this is the micropylar end. The cells which are at the chalazal end, they get degenerated. These 3 cells, they degenerate. They are getting degenerated. Let us draw it. These cells, this is the megaspore tetrad, this is the chalazal end and this is the micropylar end, and all these megaspore mother cells, tetrad structure, they are surrounded by a nucleus tissue, a nuclear cells. Now here 3 cells, 3 megaspore mother cells towards chalazal end, degenerate. That means they get dissolved, they get degenerated. Now they are all supposed to be the haploid cells. 3 of them, they start getting degenerated. Only 1 cell survives. 3 cells, they degenerate. Only one cell, which is at which end? Which is at micropylar end survives, which is as micropylar end survives. All remaining 3 cells, they get degenerated. Only one cell, this cell remain as such. Now this cell, what is happening to this cell? This cell utilizes the degenerated material of all these 3 cells. 3 cells, they get degenerated and their degenerated mass get utilized by this cell. This cell utilizes the degenerated material of all the 3 cells. Now it enlarges in its size. That means here cellular growth. Here there is a cellular growth. The cell grow in its size. How the cell grow in its size? By utilizing the degenerated mass of 3 megaspore mother cells. Now this cell is no more called as megaspore mother cell. Now, from now we start calling this as functional megaspore mother cell. Now it is called as functional megaspore mother cell. After this functional megaspore mother cell, it performs a series of mitosis now. Now, you people can answer this. This functional megaspore mother cell, is it haploid or diploid? It is the result of meiosis. That means definitely it is a haploid cell. This haploid cell undergoes mitosis. Here, complete mitosis will not take place. incomplete type of mitosis takes place. That means only karyokinesis will take place. Karyokinesis. What do you mean by karyokinesis, my dear friends? Karyokinesis means division of nucleus. Here only karyokinesis take place means in this cell only the nucleus is getting divided. No cytokinesis. No cytokinesis. That means no division of cytoplasma. Cytoplasma is not going to divide. Only nucleus is going to divide. This type of mitosis takes place. Then how many mitosis takes place? 3 mitotic divisions takes place. 3 mitotic divisions. 3 mitotic divisions. Only karyokinesis, no cytokinesis. Now this cell, I'm making it a large for our better understanding. This is the nucleus which is haploid. If haploid cell performs the mitotic type of division, then it results into the formation of haploid cells only, because mitosis is a equational division, my dear friends. Here the first mitotic division, as a result of that 2 nuclei were formed. First mitotic division, how many mitotic divisions has to take place? 3 mitotic divisions has to take place. During first mitotic division, 2 nucleuses were formed. During second mitotic division, 4 nucleuses were formed. During the first mitotic, suppose if this is the nucleus, during first mitotic division, it divides into 2 nucleuses. That means 2 nucleuses are formed. Now after this, here we'll get 2 nucleuses. Second mitotic division. Third mitotic division, from this we'll get to here, to here. That means in 3 mitotic divisions, totally 8 nuclei are formed in one cell. In one cell. Why it is one cell? Because there is no cytokinesis. There is no division of cytoplasma. Only there is a division of nucleus. As a result of that, we'll get 8 nucleated cell here. Now all these nuclei, let us label the ends here. This is supposed to be the chalazal end and this is the micropylar end. Totally we got 8 nucleated cell here. Now this 8 nucleated cell, let us enlarge this for our convenience and for our better understanding. 3 nucleuses, say for example 1, 2, 3, they move towards this end. What is this end? Is it a micropylar end or chalazal end? It is the chalazal end. Opposite end of chalazal end, it is the micropylar end. 3 cells move towards, sorry, they are not the 3 cells. 3 nucleuses, they move towards the chalazal end, and 3 nucleuses, they move towards the micropylar end. How many left? 3 here, 3 towards the micropylar end. That means 2 nucleuses, they remain in the center. This is called as nuclear arrangement. Nuclear arrangement. Arrangement of nucleus in a cell. After that, till now only karyokinesis has taken place. Now the cytokinesis takes place. As a result of cytokinesis, the cell wall is formed. The cell wall is formed like this. Cell wall is formed around these 3 nucleuses. Here the cell wall is formed around this nucleus, and around this nucleus. Cell wall is formed. But this entire is one cell. In that one cell, we have what? Two nucleuses. The three cells which are formed, towards the chalazal end, 1, 2, 3. All these three cells, they are called as antipodals. Antipodals. How many antipodals are there? There are 3 antipodals. What is the location of antipodals? They are towards the chalazal end. Now, 2 cells are formed at the base towards micropylar end. These 2 cells, they are called synergids. They are the synergids. How many synergids are there? 2 synergids are there. Above these 2 synergids, there is an actual female gamete located, my dear friends. This actual female gamete, it is called as egg cell. Egg cell. Towards micropylar end, there are 3 cells. Out of 3 cells, 2 are synergids. 1 is egg cell. Together, these 2, they are termed as egg apparatus. Egg apparatus. Egg apparatus means 2 synergids and 1 egg cell towards the micropylar end. And at the center of this cell, there are 2 nuclei located. 2 nuclei located. They are called as secondary nuclei, or they are also called as polar nuclei, or they are also called as central cell. Now, after this, these two synergids, where they are located? They are located at the base towards the micropylar end. These 2 synergids, they start developing a finger-like projections in them. A finger-like projections. These finger-like projections, they are nothing but filiform apparatus. We'll understand the function of the filiform apparatus in our future classes. They help in the double fertilization event. Actually, they help in the entry of pollen tube. They guide the entry of pollen tube. We'll discuss that in our future classes. Now, this is the structure of embryo sac. That means the female gametophyte is formed by the process Megasporogenesis, my dear friends. Now, in this mature embryo sac, how many cells are there and how many nucleuses are there? Let us count how many cells are there. Let us count here how many cells are there and how many nucleuses are there in this mature embryo sac. Let us count how many cells first. 1, 2, 3 towards chalazal end. 1, 2, 3 towards micropylar end, and this central is 1 cell. This entire is 1 cell. 2, 3, 4, 5, 6, 7. How many cells are there? 7 cells are there. Let us count how many nucleuses are there. 1, 2, 3, 4, 5, 6, 7, 8 nucleuses are there. How many nucleuses? 8 nucleuses are there. Hence, this embryo sac, it is called as 7-celled, 8-nucleated embryo sac. 7-celled, 8-nucleated embryo sac. Now, the most important point for competitive exams. This 7-celled, 8-nucleated embryo sac, it is formed by a single functional megaspore mother cell. This was formed by this cell. 7-celled 8-nucleated embryo sac was formed by a functional megaspore mother cell, which is a single functional megaspore mother cell. Hence it is called as monosporic. Mono means one. From one spore, many nucleated and many celled structure. How many nucleated and how many celled structure? 7-celled 8-nucleated structure is formed by a single cell. Hence it is called monosporic polygonum type of embryo sac. This is very important, my dear friends. Monosporic polygonum type of embryo sac development means from one functional megaspore mother cell, 7-celled 8-nucleated embryo sac is developed. This is called as monosporic polygonum type of embryo sac. Now, I hope we people have got an idea about the entire process of megasporogenesis. Now let us define what is megasporogenesis after understanding the entire process in detail. The scientific definition of megasporogenesis is the process of formation of megaspore inside the megasporangium is called as megasporogenesis. I hope you people have got an idea about the process of megasporogenesis. If you like this video, please hit the like button and subscribe to my YouTube channel for further updates. In our next video, we are going to discuss about the very important process, my dear friends, because till now we have seen the synthesis of male gametophyte, that is the pollen grain, and just now we have seen the development of female gametophyte, that means the embryo sac. Male gametophyte is ready, female gametophyte is ready. Only thing is they should mingle together, they should mix together. We'll discuss that process. It's very interesting. Stay tuned to my YouTube channel and please don't forget to subscribe to my YouTube channel. If you like this video, please hit the like button and press the bell icon for update in my YouTube channel. Thank you so much, my dear friends.